Fine. So I would say a great and warm welcome to the Pre-Antwerp Conference webinar number 3 now. And thanks to Filippo Genovese, the technical stuff seems to function, and we can start our webinar. Great to see you, but I don't see you, but you see me apparently. The webinar is called Beyond the Podium, Key Insights from the 2025 PNS Congress in Edinburgh. Now, what do we strive to? The idea is to give you an insight to what the Antwerp conference will be about. And as it is based upon the outcomes of Edinburgh, it is important to know what happened in Edinburgh. Edinburgh, the PNS, the Peripheral Nerve Society meeting, a high-level scientists and clinicians gathering every year to discuss breakthroughs in research on CMT and other peripheral neuropathies. CMT is an important part of this. And these meetings happen every year in alternating from the United States, some city in the United States, or Canada or something in America, and some of the meetings in Europe. So the last one was in Edinburgh. Bieke will mention later on the summary which Filippo Genovese, the president of ECMTF, has produced and published on the ECMTF website. So I will not go into that more deeply and not even show you the slide for it, but because Bieke will do so. But the summary which was given by Filippo is telling, and therefore I like to read this for you. CMT research is increasingly multidisciplinary and international, with tangible progress towards faster diagnosis, objective monitoring, and targeted therapies. The European CMT Federation, together with national networks, will continue to follow and disseminate these advances for the benefit of patients, families, and professionals. So that's what we are doing exactly. Now, what are the issues discussed today? Bieke will give 5 examples for what the most exciting developments reported and discussed were at PNS. And she will explain this from the scientist's perspective. Who is Bieke? Bieke Beckert, PhD, research project focused on the CRISPR-Cas gene editing. That was her PhD work. She is now postdoctoral researcher at the Peripheral Neuropathy Research Group in University of Antwerp, led by Vincent Timmerman, a very famous professor. And she also acts, that's very important today, as the PNS social media ambassador. So Ambassador, welcome. Thank you for coming and for giving us your views from the perspective of the scientists. That is then to be followed by Alexandre Oyou. He takes the patient's advocate's perspective. He is the president of CMT France. He is a board member of the ECMTF, and he is a representative of the Alliance Maladies Rares at the Foundation for Rare Diseases Scientific Council. He is also a very, very active patient advocate in many important organizations focusing CMT research, data, and all these things. So welcome and thank you for coming, Alex, for being here and giving your views then from the patient's perspective. And now, just very quickly, how should we proceed? We will first have the presentation of Bieke Beckert and try to understand what impressed her most in Edinburgh. Let us then hear the position of Alex, the patient's perspective. What does a patient, the CMT patient, think about such gatherings? Are they useful for us, the patients? What are the rules of the game today? Please keep your microphones muted, except when you are up to put a question or give a comment. Second, we would like to have a most dynamic, lively, and meaningful event. So you can put short questions and get answers, short answers, during the introduction and the presentations. But you will also have plenty of room and time after the presentations to get into the overall discussion. So if you want to intervene and interrupt, you just show your hand, and then we try to get you. But since I can't see you, I wouldn't be able to see your hands when you raise them. So I think the better idea would be that you put your question very quickly or say that you have a question in the chat. So this is how we should proceed. So I wish you and I wish us all together a thrilling and productive and delightful webinar. To start your presentation now, dear Bieke, you have the floor.
Thank you very much for a kind introduction. I will share my presentation to make it a bit easier to follow what I will say today. Please tell me if my presentation is shared.
Yes, I can see it.
Okay, thank you. So today I will talk, as previously mentioned, about some of the highlights from a scientist's perspective for PNS. As already previously said, if you want a complete overview of the CMT part of the PNS, beautifully written perspectives and highlights was written by the European CMT Federation, as you can find here. But today I will only talk about a few things just for time constraints. You can also find already the abstract supplement of PNS on the PNS website, in which you can find all the abstracts from the oral presentations and also from the posters. During my presentation, you will always see, when I talk about talks, a link to the talk specific number, and this number will be correlated with the number given on the abstract supplement. So I chose 6 topics, not 5, sorry, could not hold myself in. So I will today talk about the state of the art in gene discovery, SARM1 inhibition as a therapeutic strategy, the future directions in AAV therapy, the results from the CMT SORT trial, targeting of the neuromuscular junction, and the use of stem cell-based models to test therapies. Each topic, I will talk a few minutes about them to give you a little bit what does it mean, what was new during the PNS, and what is the importance. And afterwards, I will always open the floor for people for questions, which you can type or you can do with the microphone to really keep this very interactive so that you can immediately ask these questions. Always when I open the floor, I will say it, but you can also see then these symbols appearing. So first of all, the state of the art in gene discovery. A lot of people who get their CMT diagnosis will get some genetics involved. And as many people know, CMT is very genetically, but also clinically heterogeneous. And every year, new mutations and genes are discovered. And to date, there are already more than 135 CMT genes associated with 169 subtypes. Therefore, there is a need for a frequent update of the genetic panels. A genetic panel is a combination of all the genes and mutations, not all, but the most frequent genes and mutations that are tested on your DNA if you give a DNA sample, and then they see whether they can find which CMT type you have. During the PNS, they also said there was a need for more integrated diagnostic strategies. A big part that I found very interesting was the focus on the implementation of new techniques. And for this, I chose long-read sequencing, which is something very upcoming in the genetic field. In standard genetic panels, we use short-read sequencing. In this case, we look at a short region in your DNA sequence to see what is wrong. And normally, this is enough because when you have just a point mutation, that means one base or one letter of your DNA is wrong, looking at only a short region is enough. However, CMT can sometimes be caused by longer changes like tandem repeats or large deletions, and these can be missed with these short-read sequencing techniques. And therefore, long-read sequencing could solve this. This does not mean that long-read sequencing will completely replace short-read sequencing, but rather if they don't find anything with short-read sequencing, that your doctor could, for instance, decide to also use long-read sequencing. During the Congress, they also talked about some new genes. I did not mention them all, but you can find here all the codes to the posters and the presentation, which you can find back then in the supplement. Maybe a reminder, this talk is recorded, so afterwards you can rewatch everything that was said. So this was my first short topic. So if there are any questions or comments, you can type them now or say them now out loud.
So far, I don't see any question in the chat, and
yeah,
I do not see any hands up. So please continue.
Yeah, at the end of the day, you can also ask them, of course. So the second one is SARM1 inhibition as a new therapeutic strategy. So SARM1 inhibition is a little bit based on Wallerian degeneration. This is when your neurons are injured or you cut your neuron, that the part distal from the neuron will die. And molecular, this is because MNOT2 delivery is blocked, resulting in the activation of SARM1-dependent axonal degeneration. Because normally MNOT2 inactivates SARM1-dependent axonal degeneration. So now people thought we can maybe interfere in this process. So if we inhibit or delete SARM1, this could be neuroprotective. And indeed, SARM1 inhibition has been seen to protect against injury or chemotherapy-induced peripheral neuropathies. And there already are some early phase clinical trials for ALS and MS happening to test SARM1 inhibitors. And during the Congress, they talked about some promising preclinical results of SARM1 inhibition in CMT models, such as CMT2G, but they also talked that in some other CMT models, some CMT1A mouse models, they did not see an effect. This would suggest that maybe SARM1 inhibition will only work for a few subtypes, but also maybe SARM1 is a bit too late in the process, and instead we should target some molecules earlier in the process. I think somebody's microphone is on.
Yeah, please mute. Please mute the microphones. CF came in just now, and please.
Yes. I think it's muted.
Okay, I did it.
Thank you.
So yeah, just to summarize, so SARM1 inhibition is a new therapeutic strategy. We really have to test in which CMT types it will work. And maybe in the future, this can go to clinical trials, but at this moment, the clinical trials are only in MS and ALS happening. So are there any questions about this topic? If not, you can also ask at the end.
Yeah, I think.
Okay, then I will move forward to the future directions in AAV therapy. AAV stands for adeno-associated viruses, and these are non-pathogenic viruses that can be used as a therapy delivery tool for therapy inside the cells. So AAVs, or the viruses, consist of capsids at the outside, and depending on which capsid is at the outside, they will bind with specific cell types with the receptors. These viruses are then internalized in the cell, they can go to the nucleus, and they can then be translated, resulting in a genetic therapy to be inside your cell. A big advantage of these AAVs is that they don't insert inside your genes in your genome, and therefore they are much more safe than other previous viruses. A lot of attention lately, and also during the Congress, went to the unwanted side effects of using these viruses to deliver therapies. And these side effects are mainly immunological, and the driver was the amount of viral capsid proteins, so the outside proteins in the virus. In the beginning, there was some difficulty to deliver these tools because for peripheral neuropathies, the targets are motor neurons and Schwann cells, so these are protected by the brain nerve barrier. Therefore, the most popular route at this moment for AAV therapy are the intrathecal lumbar injection, as can be seen on this picture here. AAV therapy is mostly interesting for the clinical trials that are happening, and maybe for the people who are less aware about clinical trials, I will very shortly explain how clinical trials are divided. So a phase 0 clinical trial is preclinical. So that means that it's still what's happening in the lab, that you don't go to patients yet, just to see is the treatment useful. With clinical phase 1, you go to a small group of people and look whether it's safe to use the therapy. Most of the time, this is in healthy volunteers. Phase 2, you see the amount is increasing by going up in the phases. This will be done on patients and to see the safety and effective dose of the treatment. In phase 3, you already look at a lot of patients, and this is to see whether there's the safety, but also whether there's a benefit of the treatment compared to current treatments. And then phase 4 is the post-approval to look at the long-term effects. If you are ever interested in clinical trials, just as side information, you can always go to clinicaltrial.gov, then type in the disease you're interested in, for instance, Charcot-Marie-Tooth, and then you can find an overview of all the clinical trials about CMT, whether they're recruiting, what are the requirements to perform, and furthermore. So I will not talk about all the IV therapies that are already there, but I will focus on the peripheral neuropathy interesting. A good overview can be found on the previous webinar from this webinar series. I put the name down here. And also the AVM Teleton has written a very complete report in 2025, a bit more about the therapies that are already there for Charcot-Marie-Tooth. So also there, these will be mentioned. So now, which AV therapies do we have? The first clinical trial was for giant axonal neuropathy, in which 14 patients were intrathecally injected with AV9 for gene replacement. This 9 stands for the capsid proteins that I discussed before, which decides which cell type to target, and these are very good for the motor neuron and Schwann cells. Furthermore, AV9 is also known to be able to cross the brain-nerve barrier, so that you will see this serotype is a lot of time used for peripheral neuropathies. And in this clinical trial, we don't have that much results yet, but they already saw a possible benefit in the motor function. Another clinical trial was for CMT2S and SMART1, in which 10 patients were injected, again intrathecally injected with AV9 for gene replacement, but no results are posted yet. And the less interesting one was for CMT1A, which with 3 patients were planned to be injected, and this time intramuscularly, so inside the muscle, and this time for gene insertion, in which an AAV1 for neurotrophin 3 was going to be used. These are planned, but this trial was suspended because there were some manufacturing issues with the vectors, so they first have to solve this before moving forward. So during the PNS, there was a special keynote talk about the clinical trials in Duchenne muscular dystrophy with AAV. And these are already in phase 3. So that means that already a lot of patients were getting this clinical trial to be tested. And although the progress was promising, there were some issues with immune responses, and therefore close surveillance was needed. I want to now give a little update which happened after PNS, but I think is very important. One of the biggest clinical trials for DMT was from Sarepta Therapeutics, and these are a few months ago were paused due to some clinical problems in which I think a few deaths occurred, and they wanted to investigate better whether it was related to the clinical trial or what was happening, because of course safety is very important. And during PNS, I was happy to see there were lots of talk, posters, and experiments in which we showed that the researchers in PNS are really investigating the safety and efficacy of AAV therapies in animals. I give here a short overview, and you can see it's tested in different CMT types, different vectors were tested, and even in non-human primates. And very interesting, they even looked at the long-term safety of these therapies. So you can really see AAV therapy is a very investigated topic in our field. I also added here a few of the talks about this. So this was a little bit of a short overview for AAV therapy. But as you can see, although we are moving to clinical trials, we still have to investigate quite a lot of things. So are there any questions about this? I will think you will...
I don't see any question now.
The questions can also wait to the end of the presentation.
Yes. I think we wait for
I think.
presentation. Yeah.
Yeah, so the next one are the results from the CMT SORT trials. Probably a lot of people have heard about these. So CMT SORT is the most frequent recessive form of CMT, which is an axonal form, and was only discovered in 2020, so quite recently. And this is caused by mutations in sorbitol dehydrogenase. So which pathway is this important? This is important in the glucose to fructose pathway. So normally glucose goes to sorbitol through aldose reductase, and sorbitol goes into fructose through sorbitol dehydrogenase. And people with CMT sort have mutations in the sorbitol dehydrogenase. And this will result in an accumulation of sorbitol, which is neurotoxic and result in neuromuscular deficits. Therefore, a clinical trial was started, the INSPIRE CMT SORT trial, which is already in phase 2 and 3, by Applied Therapeutics. And they give the drug govorestat. I don't know whether I pronounce it correctly, but this is an inhibitor for aldose reductase. As you can see here in the pathway, aldose reductase is an enzyme before the sorbitol, and this could reduce the sorbitol accumulation and therefore alleviate some of the side effects. So very interesting, during PNS, they gave some clinical key findings from 12, 16, and 18 months after the start of these treatments. And first of all, they saw that the blood sorbitol level already decreased after 12 months, which was quite a good sign to see. The primary outcome of the trial was normally the 10-meter wall current test. And unfortunately, this was not significant at 12 months and therefore was removed from the CMT FOM composite score. However, the CMT Health Index improved after 12 months, which are the patient's reported outcomes. And also very importantly, they saw through the MRI measurements that the muscle fat fraction decreased at 24 months with a significant change. This means that the potential at 24 months, the disease progression starts to slow down, which is a very important finding. At this moment, they're still analyzing all the rest of the data because, of course, a lot of data has been created. And their goal during the PNS with this was still to get FDA approval in 2025. But I do believe that maybe this will be a little bit delayed, but you can find more information about them on their website. So the CMT SORT trials were very interesting and very encouraging results. And I'm looking forward to see a complete report about what is happening. I will maybe just move forward, but you can ask questions at the end. The next one, this PNS, it was very remarkably that the targeting of the neuromuscular junction was quite an important topic. The neuromuscular junction is a connection point between your neurons and your muscles. Here I zoomed in at the right, and you can see here in yellow the neurons, and in pink you see the muscle. And you see them connecting. So normally when the stimuli by the nerve comes inside, this will result in an action potential which will release acetylcholine. This acetylcholine can bind with acetylcholine receptors on your muscle. This results in a positive stimuli and the activity of your muscle. So this is a very important function to have normal muscular function. And it is known that neuromuscular junction is dysfunctional in CMT, but quite understudied. And also something that I found interesting during the PNS, that they have seen that the neuromuscular junction decline in aging seemed quite similar to CMT in mice. Also, a big part of this topic went about what NMD Pharma is doing. And first of all, they performed an observational study in CMT1 and CMT2 patients in which they really looked into the neuromuscular junction dysfunction, because although we know something went wrong, it was not really studied at this moment. And this was the established CMT study. They saw that the neuromuscular junction transmission was dysfunctional. They saw, for instance, an increased level of jitter and neuromuscular junction blocking. They also saw it was worse in CMT2 patients than in CMT1 patients. I maybe didn't mention, but they used different subtypes of CMT1 and CMT2. So they went as a general thing that they went through this. And they also saw that the level of dysfunction correlated with the disease severity. So based on these results that were talked about 2 years ago, they now started a clinical trial with a specific drug. And in this drug, they targeted the chloride ion channel, which is a skeletal muscle specific channel. I previously already talked about the acetylcholine receptor, which was important for the positive stimuli, but the chloride ion channel is also present, and this normally results in an inhibitory signal to make sure that your muscle response is not always active. So their idea of NMD Pharma was to add an inhibitor for this chloride ion, and therefore they could enhance the neuromuscular response. They started already a phase 2A clinical trial. At this moment, they're still recruiting patients to look in the efficacy, safety, and tolerability of this drug for different subtypes of CMT1 and 2. So at this moment, there are no results yet because they're still performing and recruiting patients. They already applied this drug in myasthenia gravis, which was their first target, and they saw an improvement in muscle strength and an enhanced neuromuscular transmission, which was quite promising. They already have some preclinical data in CMT mice, both CMT1A and 2D, and there they saw that there was an increase in muscle force after application of this drug. In CMT2D specifically, they saw also an improved motor nerve function and a better preserved neuromuscular junction integrity. It is important maybe to remark that this drug would not solve the cause of CMT, but would be a symptomatic approach to reduce the neuromuscular deficits. So this was a little bit an overview of this neuromuscular junction, and I think in the coming years we will all look forward to see the results of the clinical trial. And I will now end with my last topic, the use of stem cell-based models to test therapies. As you already heard previously, a lot of the therapies that we talk about are first tested in animal models to see whether they're safe and efficacy. But animals are not the same as humans, so they cannot completely mimic. And that's maybe also one of the reasons why a lot of therapies that are working in animals are not working in patients at the end. Therefore, there is a need for human-derived models, which could hopefully mimic better what is happening. And in a lot of cases, researchers use now induced pluripotent stem cells of IPCs. And IPCs are derived from a patient, from a blood sample, a urine sample, or a skin biopsy. And because it comes from a patient, they will have the same genetics as a patient. So if you have CMT, also these mutations will be present in the IPCs. And these IPCs can then be matured to motor neurons, Schwann cells, to different cell types. And these can then be cultured in vitro in a dish. And they already seen when you have IPCs with a mutation that they show CMT characteristics. So it's very interesting to investigate CMT, but also drugs on these cells. But of course, CMT is not caused only by one cell type, it's caused by multiple cell types. For instance, with CMT1 or the demyelinating forms, the motor neurons, but also the Schwann cells around the neurons are very important. Therefore, you can also make co-cultures in 2D. But at this moment, the co-culture of the human co-culture of motor neurons and Schwann cells is suboptimal. Also, you're still working in 2D, which is not the same as the 3D situation in which your cells come into contact in 3D with different cell types. Therefore, a few years ago, people started to make 3D models. And from the group of Vincent Timmerman, my own group, we published about the 3D neuromuscular organoids, in which you could see myelination happening inside these spheroids, these organoids. And also when CMT cells were used, we could see deficits in the myelination similar to what we saw in patients. During this congress, myself presented a new model, the 3D neuromuscular assembloids, in which a motor neuron spheroid was fused with a muscle spheroid. And the good thing about this is that you really can see the directionality of the neurons going to the muscles, which makes it easier to investigate what's happening. This picture was also during the PNS chosen as a ePNS cover, which is the journal affiliated with PNS. Another part in the Congress is not only IPCs could be used, but also a research group talked about that dental pulp stem cells could be used by the group in Hasselt, by Professor Wulfs. And they saw that from the teeth, you can derive dental pulp stem cells from patients and then also make the motor neurons and Schwann cells. So this is a new source of cells that can be used. Overall, all these models are very interesting as a tool to test therapies on, and hopefully these will mimic much better what is happening. So this was a short overview. Again, a bigger overview can be found on the European CMT Federation, and also quite certain that a lot of the topics that I talked about today will come back during the European CMT Specialist Conference on Antwerp. So if there are any questions, please let me know.
So far, thank you very much, Bieke, for this wonderful introduction. I mean, it was so much of information, so it is not easy to have questions. We need a little bit of time, perhaps, to get into our brain what you tried to put in. And yeah, I have a very simple question at this moment. But this is not important because I see a question of Vincent Timmerman. It is very possible that someone diagnosed with CMT1A duplication can also have a point mutation in another CMT-associated gene, but this will rather be exceptional. Vincent, would you just talk and give your comment and ask your question?
I think it was a reply on a previous question from Simone Lucante.
Yeah. Okay. Do you have a comment on this, Bieke?
No, maybe for the people who could not read the questions, the question was asked whether people diagnosed with CMT1A 20 years ago, whether there was any chance that with the most recent gene-based diagnosis, an other type could be revealed. And Vincent Timmerman, who found the CMT1A duplication, has said it's very possible that someone diagnosed with CMT1A duplication can also have a point mutation in another CMT-associated gene, but this will be rather exceptional. So this was a question about what this was.
You can open your microphone, Vincent.
Vincent is trying to open the microphone, but it doesn't work. Could Filippo help? I try, but
the mic is on. I mean, they can put the microphone on.
Vincent, try to put your mic on.
Yeah. Does it work now?
Okay.
It's working now.
Sorry, I had a problem to put the microphone on. Yes, indeed. So you can be diagnosed with a duplication recently or many years ago, and that doesn't exclude you from having other mutations, of course. Maybe not related to CMT, but can also be other diseases. So that's that's what they call a double hit, which is not always nice to have. But everyone is prone to have mutations, every one of us. So not only for CMT, but for any other type of disease. So I would say it could be, but it could be rather exceptional. But that depends, of course, and needs to be a careful diagnosis. I cannot say more than that. But that can have an effect also on the treatment, of course. If you have a treatment for CMT, you have to be aware that other disease might not be treated for the same treatment as for CMT, and the other way around. So if you are treated for cancer, that doesn't mean that you will be treated for CMT. So every disease will be very carefully looked at to see if a therapy would work, yes or not, for an individual patient or a family. Hope that's clear. Thank you.
Thank you very much. Are there other questions? Stefan Will asks.
Yeah, I can also read out. So I've got recently diagnosed with CMT1X. I just wanted to know, like, I see all this research, and so I was surprised also to see that there's almost 170 subtypes. I was not aware of that. And wanted to know, is there like a priority call, like which CMT types are addressed first in research, or because for me it's a little bit, there's a lot of information, but I'm trying also to digest what's available for specific types, like my type, CMT1X, for example.
Mm-hmm. Maybe start. In research, we really investigated a lot of types. I know a lot of researchers who are investigating CMT1X. Of course, CMT1A is maybe a little bit more investigated because the main amount of people have it, but it does not mean that we are only looking at CMT1A. Also, CMT1X is also very investigated. I do think multiple groups are maybe focusing on some CMT types more specifically, but I think CMT1X is definitely also investigated.
Okay, thank you. So there's not really a priority call. It's more like go with the flow with the research and then see where it works.
Yeah, and I know in our group we investigate multiple CMT types, and in most groups they will investigate multiple CMT types. So it's not that only one type gets all the money. Honestly, in my own experience, all the CMT types are getting the same amount of attention from governments-wise.
Okay, thank you.
Yeah, would be a very strange political choice to say these types are now preferred and money is given for this and not for the others. But it's a fact that CMT1A is the most frequent type, so it gets the most of attention, I guess. There was a question if there's any news on FarNext and the PXT 3003. Do you know anything about this, Bike? It's not exactly the subject of PNS, but
I think it was also discussed a little bit. The FarNext trial, PXT 3003, was a trial that a lot of people hoped was going to be working and was investigated. It was quite successful in mouse models, but unfortunately, when they went to patients in clinical trials, the results were less good than expected. I know in the last webinar of last time they went quite deeply into this, but to my understanding, they are still looking to maybe do a follow-up. I don't know whether it's FarNext themselves or other related companies, because they were maybe afraid that the sorbitol that was injected was crystallizing, and maybe the sorbitol, which was one of the 3 components that was injected with FarNext of the PXT 3003, was maybe a problem. So I don't think they're completely thinking of putting its weight as opportunity, but unfortunately it was not as good as expected. I think that maybe somebody else has to say something about this.
If you want, I can say something. This is Angelo speaking. Because we have been involved in the trial, and to the best of my knowledge, the trial has been stopped for not having any result. And actually, I think that the company is not existing anymore, because they had, problems with the administration and money and so on. So, I mean, again, to the best of my knowledge, everything is stopped and there are basically no results.
Thank you, Angelo. Except that there's some news, Mary Kate reports from the PNS, yes, that there was the Chinese trial is continuing apparently, and we don't know what is coming out of this.
Yes, so, exactly. The thing is, Angelo is right. The company in France, FarNext, is done now, and the results in China were showing, and there were a poster about that with some results, but it's not very clear. It is not very good results. It's not evident. That's it. That's just what we can say for now.
So thank you. As you are talking already, I think we switch now to your patient's perspective for a presentation for the PNS. What is your experience? What has fascinated you most, and what are the remaining questions after this conference? You have the floor.
Okay, thank you. Good evening, everyone. And thank you, Bika, for this brilliant feedback on the highlights of the PNS. I would say just before attending a scientific conference like the PNS, you have to learn 2 languages, the English and the researcher language. You can feel it today. It's not easy topics, but it's very passionate. So for my part, I am here today to share a patient perspective. So I am Alexandre Rouillot, President of CMT France and member of the European CMT Federation, but I'm also, and above all, a CMT patient, as well as my 2 children, Clara and Sasha, aged 9 and 14. But before becoming involved as a patient advocate, I studied science and obtained a master's degree in logistics and IT. So I worked in this field. And then I became CEO of a company in logistics and transportation. I ran 4 years. It was the highlight and fascinating activity, the highlight of my career, but I had to stop in 2022, mainly because of my CMT. It wasn't an easy time, to be honest, but it was also an opportunity for me to make sense of everything that was happening to me and to my family. So after a long reflection, few weeks, few months, I jumped into the fight with my entrepreneurial spirit, with a clear goal. I wanted a cure for my children's generation. So today, with this goal high on the list, I share CMT friends, and I joined major organizations such as ECMTF, the Rare Disease Alliance, Rare Disease Foundation, the FM Teleton, and I participate in major scientific conferences on CMT, such as the PNS. And so I'm here today with you, and I want to say today it's me, but it could be you. I know there is patients in this attending this webinar. It could be you. I mean, all the patients have a role to play at every level. We really need engaged patients, and I wanted to say that today. To return specifically to the PNS, this is a scientific conference, of course. The idea plays to learn about the latest international research in the field of peripheral neuropathies. So the program is therefore structured around a high-level presentation, some highlights of which Beaker has just presented with great clarity and expertise. Thank you. You can see that there is no slides. Beaker, you worked a lot. Thank you for that. So on my side, I just want to speak more about what represents the PNS for me and for patient representative, and what's happening behind the scene. So the PNS for me is firstly a community. It's just not a scientific conference. It's a huge community of patients, researchers, clinicians, and pharmaceutical companies that come together every year, strengthening the links that make true collaboration possible between all these stakeholders. This spirit of community is not just important, this is vital. It gave us strength, trust, and it reminds us that we are not alone in this journey. Every link is crucial, and placing the patients at the heart of this community is essential to me. So just for example, this bridge, I mean meeting patients, is very impactful for researchers. You know, unlike clinicians, the doctors, most researchers have little or no contact with patients in their daily work. Much of their work happens at the bench with complex, repetitive, sometimes frustrating tasks. I think Beaker can confirm it. So when they connect with patients, they can see the meaning of their work more clearly. Why it matters, who is it for, and how it can change lives. And this human connection is essential for motivation and long-term commitment. I heard that from so many researchers when I meet patients. It's very impactful. Now, you know, the researchers need patients, and patients will need researchers. So research is the first step toward the treatment. Sometimes it's abstract for us, you know, the patient. It's abstract. There is no impact in our life directly. But without research, there is no clinical trial. This is the first step. Clinical trial, and without clinical trial, no drug. That's simple. We do need research. And I can assure that there has been huge progress in research on CMT during the last years. I attended the 3 past PNS, and I can confirm there is progress. Even if I must admit also that the road remains long, and that from a point of view of a patient, it doesn't go fast enough. But every year, there are many steps forward for on any CMTs. We saw that during the presentation of Baker. With enough proof of concept on the research, biotechs and clinicians can sit around the table, and patients, of course, and move towards clinical trials, and an essential step in showing whether a drug is effective or not. And if I speak about clinical trials, because for me it's the step very close to the treatment, we saw different highlights from the PNS, and I want to speak. The most important highlight for me is CMT sword with applied therapeutics. Why? It raised a lot of attention. We saw that there is good results in these clinical trials. It's very clear, and we need this. Even if it's a drug for a specific subtype, firstly, there is a lot of people affected by CMT sword. And from my personal perspective, it's very good news if there is a treatment for CMT sword for all the community, because we need all a victory. I mean a drug on the market, and we have good reason to believe that this one could be the first in the market. It could pave the way for the other treatment, to prove it's possible to treat CMT. So we need a victory. I want to speak about also NMD Pharma clinical trial, even if Baker spoke about that. This clinical trial is running in US and EU. It's an interesting approach in the sense that it could benefit to most of the CMT and improve our quality of life. So there is a lot of hope in this clinical trial too. Another very promising clinical trial on CMT with MSCs, mesenchymal stem cells, with N cell in South Korea, which have good results on CMT1A and CMT1E. And this cure could benefit also many subtypes. So it's very encouraging too. And there is a lot of promising therapies currently in development. This gives a real hope that treatment will arrive in the coming years. Okay, the road is long, but we can have rational hope about that. So I won't go into further detail into the science because Baker presented this very clearly. And I just want to say that if you want to go further, there is a booklet produced by the AFM Téléthon every year, the advances on CMT 2025 this year, available soon in English. It's already available in French on the website. And I have the honor of validating this document, and it offers a great overview and help to appreciate the progress on CMT every year. I think I will put the link in the chat after. And to conclude, I would like to share encouragement. This webinar is just one step before the Antwerp CMT Specialist Conference, the second one in Europe. And the program has been co-designed with patient representatives alongside clinicians and researchers. For me, for all the reasons I said before, it's very encouraging sign. It sets the tone for a conference that will not only raise the quality of the dialogue, but also ensure that the outcomes have a real impact for people living with CMT and related neuropathies. So, last sentence for me, after that we can discuss. The science advances because of brilliant ideas, but progress becomes real when patients can feel it in their daily life. Our job is together to close this gap.
Thank you very much, Alex, for this very fascinating view, which is governed by the idea of hope for the patients, of course. And the question of the patients is always what all these researchers and scientists are doing. What does it help me as a patient? Of course, that's the patient perspective, but you brought a lot of other interesting aspects into this discussion, and I thank you very much for your contribution, as much as I thank to Bieke that she introduced us so clearly and so competently into the field. I had one or two questions from the last part, so on Bieke's introduction, but the second one is also for you. So the question was, were there any discussions at the PNS about this CRISPR perspectives for further research, for finding a treatment, etc.? Can you remember? Can Bieke remember? Can anybody remember what was about?
I do remember.
CRISPR? Sorry?
I do remember CRISPR, which was also my topic previously, so I always pay attention to it. Specialist. CRISPR, most of the time, was used to make the stem cell models that I remember, so which you make in the IPCs that you can induce the mutations. But there was one talk about the amyloidoids, which they talked about phase 1 clinical trials, but I think they only look at the safety mostly, I think, so I don't really remember the results, but you can definitely find it back in the supplement of the PNS. But so there was, I think, one talk in CMT about this. In amyloidosis.
I attended, sorry, because to interrupt you. Yeah, I saw this presentation. It was very exciting because this is the first time we are using the CRISPR in vivo on human. And the results were quite good. I think I could ask you, it was Andoni Echaniz Laguna who presented this, and maybe we can ask him to share the presentation. For me, it was exciting because it was the first time with safety and good results with people with amyloidosis.
Amyloidosis, yes, it's a difficult word.
Yes.
Yeah, thank you. So, and there was another question, and it was Walter who raised the hand.
¿Lo has escupido?
Who?
¿Qué?
Who is this?
I muted her. Yeah.
Muted. Yeah. I see the hand, one hand, but I can't discover who is it. Whose hand?
Actually, 3 hands.
Filippo.
The first hand was raised by Walter Pernice.
Ah, yes. I haven't seen that. Walter, are you here? You can unmute.
Well, he probably raised his hand by chance, by mistake. So there's also Simone who wants to make a comment. Please, Simone.
Yeah. Hi, everyone. First of all, thank you for this. It's quite nice to see a community, so people don't feel alone in this, I would not say battle, but like this condition. From a patient point of view, I would say that it's really amazing. I agree, like, we need victories. It was a beautiful phrase, like we need to demonstrate that this can be treated. But at the same time, I want to add something from a patient point of view. What I experience for myself is that the CMT since affect the whole body. An holistic approach is also good because for 10 years, my disease was quite stable. Then I had a year and a half of poor diet, poor sleep, insane amount of stress due to job, insane amount of stress. And in 1 year and a half, I saw the worsening of my symptoms. that I've not seen in 10 years. So I think it's important to also, find new therapies, but at the same time, it's important to highlight how I really, it doesn't seem, it seems new age, but, meditation, good diet, avoiding a stressful life. I think those are quite important as well.
Yeah, definitely. I mean, CMT is not disconnected from our whole body, so it's part of it. So as long as we don't take care of ourselves, we will definitely get things worse and worse. So I totally agree with you. And so healthy lifestyle, not gaining weight is also sometimes important, because your bones and muscles, they are still suffering from not coping with your poor nervous transmission. So if you had weight on the top of that, then things can only get worse.
Yes.
I have a few comments and a question for BK. Is still
Yes, please go on.
Online. She might not be. Oh, there you are. Yeah, correct. I'd like to ask you a general comment, not on a specific topic, but on the science you saw on the PNS compared to the last edition, whether you saw a significant increase in the type of science that was produced, the quality of science that was produced.
The question is about the lot of science that is happening. So,
well, yes, a personal comment about what you see and which is your impression, your feeling compared to, let's say, 10 years ago.
I will be honest, 10 years ago, I did not know what CMT was.
Yeah, too young.
Maybe a little bit, but already in comparison, I think I'm almost a bit more than 2 years in the field. I have the feeling that more and more people are also getting funding for CMT research and also are able to investigate, and also more and more different fields are being investigated in CMT. For instance, the neuromuscular junction, a few years ago, you could not find almost anything back about this. Well, now this is being investigated, and with the MD Pharma now investigating, this could potentially lead to a new therapy. So I really think we are moving forward, and we started to become a bit more creative and searching in different ways. So I really think the field is really evolving. I think maybe if Vincent is still online, he will maybe know better, because he's been a long time already in the research field, but I think there is a lot of evolution happening in the research field.
Yeah.
Yes, I can confirm, absolutely. Yes. This is, my first PNS meeting was together with Angelo, who is also here, I think, and that was somewhere in Italy, in Rapallo, if I remember.
1992.
1992.
Some of the people
indeed.
who are here were not even born.
Yeah, so I was there in 1992, and Angelo also. And yeah, then I think we were maybe 80 people maximum, something like that. Now we at the PNS in Edinburgh, we were reaching, I don't know, above 1,000 people. So just to give you an idea on the number of...
Vincent, you described for the first time the duplication.
Yeah, in 1992, yes.
You brought us the news about the duplication at the meeting in Rapallo.
In Rapallo,
the
indeed.
Meeting was, as far as I remember, were about 160 or 200 people altogether, but working on CMT, really few. Yeah. I was already working on CMT, and our geneticists were already working on CMT. And Mary Riley was there. She was quite like a student or something like that.
Yes, she was a student.
She always says that she was a student. But of course, there is there was, enormous work done in these years, particularly in the last, say, 2 to 5 years. And it goes along with the findings in other hereditary, myopathies, motor neuron disorders, and neuropathy. So, it's following the flow of knowledge regarding other genetic neuromuscular disorders, I would say.
Yeah, and also technology has evolved dramatically.
And technology is very much evolved.
Yeah.
Exactly.
So after all, after the Edinburgh meeting of PNS, what is your expectation? When will we have the first treatment on one of or more of the types of CMT? Is there expectation? I mean, for the patients, there is so much hope all the time. And this was since many years already, much hope, little promises, but be careful. Is this changed? Is there a game changer somewhere, technology, AI, or what could change the situation, except that more people now are engaged in research on CMT? So perspective, when will we have the treatment?
If I can answer, I think that Simone, he said important things about that. I mean, the holistic approach is very important. We see patients, I mean, we started our CM, as Filippo knows, 21 years ago, we started with our CMT clinic here in Genova in 2004. And we are following patients since then, which basically didn't have very much of a worsening. They're pretty stable. So it's very important to tailor the therapies almost for each patient, not for all CMTs. Like I was saying when I wrote the notes about the survey, there is such a difference between the different CMT patients. Not talking about amyloid neuropathy, which is a completely different disease and field. But I think that first of all, you have to take care of the patients, see them every 6 months to 1 year, and really measure the disability of the patient. And then have this holistic approach, and then tailoring specific therapies for specific patients. For example, inflammatory superimposed inflammation. Sometimes it happens. And so the patient has to be aware of that. So a worsening of any type of CNT in 1-2 months need to be evaluated and maybe treated with simple therapies like IVIG or steroids or whatever. So it's very important to follow up the disease and then decide which is the good time to start with the therapy. Of course, there are already some therapies like the SWORD, related CMTs. So with aldose reglutase, I don't know if it will work. We will have to see, if it will work. But there are some approaches. But my view is, I don't know if Vincent agrees with that we have to follow very, very closely the patients and understand when this is the worsening, and especially, have really tailored therapies for the different patients.
Yeah, thank you. Yeah. So natural history may be an important thing and much more detailed than we normally used to collect that. And the study, when I understood Simone rightly, certain life conditions, outside life conditions, and I have the same experience myself, can have a very strong impact on the progress of the disease. In my case, for instance, I can report each time I moved from one city to the other, from one place to work to the other place to work. Each time there was a huge step forward in the progression of the disease. So I decided not to move anymore. But this is not a therapy, of course. But studies on that, on the natural history, will be most important. Now I have a question. Now Walter has put his question on the chat. And the question is, I guess, to Bieke. because Bieke talked about the stem cells, Walter says, how can you get the stem cells from patients to study on that? I think you mentioned the teeth, the wisdom teeth approach, which Esther Wolfs is following, but you can perhaps tell us a little bit more about this.
I think, or he's asking about the therapy, the mesenchymal stem cells. Maybe I will also talk about that. But for research, you of course have from the dental pulp stem cells, like you said, from Professor Wolfs. But IPCs are just cells from a blood sample, a urine sample, or a skin biopsy. We can derive normal cells from these, and we can then transform these cells to these IPCs. If you talk about the mesenchymal stem cell therapy that Alexandre talked about, these were, I think, most of the time from Wharton's jelly, from the umbilical cord, but theoretically, mesenchymal stem cells can also be derived from the bone marrow or from adipose tissue. But I think in the clinical trials, they're using from the umbilical cord. But I think maybe Alexandre will know about this more because it was in the report of AVM.
Alexandre?
Yes, sorry, yes. About MSCs, you mean? Yes, it's a therapy it is not stem cells, in fact. There is a confusion between stem cells and mesenchymal stem cells. The stem cells are not replacing other tissues or cells, but they are secreting some molecules to help for protection of the axon and myelin regeneration. This is not stem cell changing for the cells, you know. It's not programmation of cells. So that's what I understood from this therapy. And it's really crucial to understand that because it's about safety. Using stem cell, injection of stem cell, you know, it can cause cancer, some issues. But with mesenchymal stem cells, there is no problem on this side. You could have immune reaction. That's the only issues I understood from this kind of therapy. And what is exciting and we need to be interested in is that the results in South Korea on CMT1A, even if it's not a lot of patients, CMT1A and CMT1E, the results are very good. It's 12 patients to my mind. Yes, that's it. And the results are very interesting. So we need to understand why, how it works more, and this. We don't have a lot of clinical trials with good results, so we need to keep our eyes on this.
Okay.
And they will present at the Antwerp conference. They will present it, right?
Mm-hmm. Alex, that would be a question from my side when you reported of the Korean project or the Korean approach. Could you go into that a little bit, what you know about this? Because was it discussed in Edinburgh or is it in another project you talked about?
They already presented something in Montreal. And they presented new results in Edinburgh. There are posters also, and I met them with clinicians, and they want to understand more what does it work. I'm not a clinician, I'm not a researcher, so I just explained that there is a clinical trial. It's a spin-off. In fact, Ansel is a CDMO, so they are manufacturer for mesenchymal stem cells. And it's a spin-off of the Samsung Medical Center. So it's serious company. And the thing is, the trait, okay, Filippo, thank you. Filippo put a link into the chat. You can have a look at this. And the thing is, the results on CMT1A and CMT1A patients are very encouraging. So the thing is, I think the researchers and the clinicians must have a look at this, because we don't have a lot of great news like this. That's what I'm saying, and I'm looking forward to seeing that during the Antwerp conference. And if you want to know more about that, come to the conference.
You are an excellent advertiser, and thank you very much for calling for. Registration is still open. We extended the deadline a little bit to allow others to join the conference, and please make some propaganda everywhere so that we get the room good filled. But now let's come back to the questions. I have got a question from Karen Hartford, who asks, would anyone have an update on NMD Pharma's pediatric investigation plan? PIP. That's the abbreviation. NMD Pharma's pediatric investigation plan. Is there anybody, Vincent or whoever is here present at this wonderful meeting, to talk about this? Or perhaps you, Alex? Or perhaps
if
Peter?
I can add a comment on that.
Yeah.
Usually this kind of studies is addressed after you have a very strong scientific evidence that your drug is working in adults. So I guess they will schedule it, hopefully, on later stages. So we have to wait. And we have to, we don't have to rush when we're talking about clinical trials. I mean, we patients and also clinicians and researchers, we want to be sure, 100% sure that the drug is safe and it's not harmful despite being effective. So we have to be cautious with clinical trials. I understand that you, Karen, are the mother of a 5-year-old with CNT. Most of us, I mean, patient advocacy group representative, just like me, Alex and Ingo, we are CNT patients too. So we understand what you're feeling, but yeah, we want to make sure that first of all, the drug is safe.
Yeah, and the second would be nice if it were also efficient. Yeah.
Yeah.
But of course, that's too much to hope for the moment. But let me come back. Thank you, Filippo. Let me come back to the introduction when Bieke talked about the NMD Pharma 670 trial or idea. Yes, it's the junction between nerves and the muscles. And if this junction functions better, then that could be a lot of hope for all the patients, whatever type of CMT we might have. Just my question now is, I do not really understand. If my nerve is dying because of whatever mutation in the genes exist, if the nerve is dying, then of course there will be no impulse anymore to go through the junction to the muscle. So would that really, a drug which helps the junction to function better, would that really help? So you said it's not a causal cause, the cause is not attacked, but it's a symptom of the of the disease, which is the attempt is to ease that, to improve the transmission of the impulses from the nerves. But if the nerve is dying, would that help? Is there anybody? Beeke, can you explain? Am I wrong with this question? Or
this question was also asked to NND Pharma during the Congress. So they also asked them, what if all the nerves are dead? Can we still do something? And they indeed discussed that the phase when the drug will work best will also be something that needs to be investigated. And of course, if all the nerves are dead, maybe it's too late. So, but they indeed are still investigating a lot about these topics because their drug was first investigated or really made for myasthenia gravis, in which the acetylcholine receptor, the positive stimuli receptors, are getting lost, and only the inhibitory channels are there, and there is like the inhibitory effect is too much, and that's why they want to inhibit. And they now wanted to also try to use it in CMT, and they saw in their mice model that there was a positive effect, so they continued. They also think maybe will it be better if there are already the neurons and muscles are still completely okay? Will it not work anymore if the neurons are completely dying? These were all things that they say they will investigate in the future. But I think even in CMT, even at the end, there will still be some neurons working, some muscles where there can be an effect. But it's something they are aware of, and they really are investigating at this moment which stage will be the best.
Thank you. Yeah. Can I ask another question? Until questions from the audience are coming.
There are two questions waiting, actually, but yes.
Are there? Where?
Yeah, I've raised my hands 20 minutes ago.
Go on.
My arm is getting tired of staying like this. Anyway, yeah, it was just a comment to thank Beeke for the nice presentation, and as well as Alex. And to add to what he stressed about the collaboration between PNS and patient advocacy group. PNS is also a chance of networking with other patient advocacy groups. So I see there are many patient advocacy groups representatives in the chat. So we had the chance to interact with, of course, the European CMT members, as well as Mary Kate and other from the other foundation and organization. And we are trying to make this interaction with PNS more and more effective, interacting with all the patient advocacy groups. That's it. There was also a question by Manuela.
So Manuela, where is she?
Manuela Timmer. She might not be in front of the computer right now. So perhaps you could make your comment in the meantime. Ingolf.
Yes, yes, my point was the observation is that if you do exercise and you exaggerate, then there is the progress of the disease is stronger, is quicker. So it was also observed in the rehabilitation clinic here, where I was for 6 weeks 2 years ago. And that seems to be a general observation that if CMT patients do exercise and training, and if they move regularly, smoothly, and not too much, then that's useful. But it could be a problem and could increase the speed of the disease progress if you do too much. So that would mean that if the nerves are excited too much to press the muscles to work, then the progressive dying of the nerves is more rapid, as if you do this smoothly and the exercise not too much. So is there any research on that? Is there an explanation for this? Why is it bad to exercise too much?
Ingolf, sorry, because I have to leave, in really few minutes. We did publish a few papers regarding the so-called overwork weakness, which is a reality in CMT patient, but not only in CMT patient, like, for example, in ALS and so on. It's not a problem of nerves. It's a problem of a muscle which is denervated, and so is not able to have too much exercise. For example, we compared the right with the left hand. The right hand is used more than the left hand in most people, And as a matter of fact, it goes, there is a much more, it's worse in the patient, the right hand than the left one, because of this, because of a work weakness. So what I would suggest is to work out, but not beyond the level of fatigue. So when fatigue arrives, then you have to stop exercising.
Okay, yes.
And that's an important point. It goes along with what Simone was saying. It's not just, a drug or a molecule or something like that. It's a way of speaking to the patients and discussing with the patients the best approach. Exercising is good, but it does not have to go beyond the fatigability. So it's really something scientific. This is scientific. Unfortunately, I have to say goodbye to everybody because I have my daughter waiting for me. So bye-bye.
Anyway,
we can discuss again maybe in Antwerp, okay?
Thank you, Angela.
Bye.
Bye-bye.
Thanks for joining.
Thank you.
I've heard that we shouldn't overexhaust yourself with CMT. I actually train quite a lot, so between 6 and 8 hours per week, and I spend like several hours on the bike per day on my vacation. And I've actually noticed that it does me I do pretty well after that. So for example, I have vacation trips where I spend like between 6 and 7 hours on the bike per day for a whole week. And yes, I'm exhausted at the end of the day, but I still can get up the next day, get on the bike, and do another 6 or 7 hours on the bike. So that's why I'm a bit concerned. Am I overdoing things? But in overall, like after those times that I really exhaust myself, I actually walk better. Like I feel like my like the whole stability is better, and the nerves seem to react a bit better. So is it maybe that certain types react differently? I also don't want to do anything wrong, but I really feel like extreme exercise kind of helps me.
Yeah, that's exactly what Angela was saying. Every one of us is different. We all have our own threshold. So we have to learn where is it and to not overwhelm our body with exercise.
Okay, so I guess then I have to figure out when I'm overwhelmed, because so far I've not reached that level yet.
Apparently you are in a very good shape. You are still fit, and if your limit or your level of overworking, you do not reach it. That might happen, and therefore it's good. You should continue as you do. My question was a little bit to the researchers. Is there research on an explanation? Why is it disaster, or might it be a disaster for the muscles or the nerves or whatever, for the system, if I overwork it? If I have CMT, others who overwork have some pain in the muscles the day after and have some problems, but then it's over. But with the CMT, it seems that the nerves are then dying if they are overworked. And why is it?
Ingolf, if I
Yes.
can just react to this. I think we should be careful about this message, because there is a lot of papers also, and we can share it, showing that doing exercise regularly is very beneficial for the patients. And on the other hand, not enough exercise, sedentarity is very bad for us. And I know, because I'm fighting for that many patients with CMT are not doing enough exercise. That's the way of life today, you know, computing, the car, that the way of life for every people. It's easy to say, I have a CMT, I don't want to move too much, and I need to take care about my muscles. And it could be very bad for us. So we need to take care about that. Okay, not too much exercise, not to go too high, and take care, learn, and we need to hear our body, and to work with physiotherapists and coach, and not alone, but do exercise. I want I need to say that do exercise. It's good for us. There is many papers showing this on CMT patients, and also on Duchenne patients, people with muscles disease. So doing exercise is very good for us. It's important to understand that.
Okay, thank you very much. It's almost the concluding words. Alex, thank you very much. Time is over now, and some have already left and have to leave us. I would like to conclude, and first of all, thank all the participants, everybody who joined us to listen what happened in Edinburgh from the different perspectives. And then I would express my gratitude already now for those who sponsor not only the conference in Antwerp, but that includes the pre-Antwerp webinars. And this is, now I have already 6 names who I can thank and quickly mention. That's the EU4Health program, which gives us enormous, generous funding. The AFM Téléthon from France, which gives us lots of money, and Applied Therapeutics, the company who is leading the SWORD trial, it supports us very considerably. Then Ulysses Neuroscience, and then the Flanders State of the Art and the Flanders Research Organization, the two, all support us very generously and help us make this webinars happen, and also to organize the 2nd European CMT Specialist Conference. That takes the 23rd to 25th October. Come and join us, and let's continue the discussion if you can. And for the neurologists and the CMT specialists who have not yet registered, please, the registration is still open until the 26th of September this year. Thanks again very much to Bieke for this wonderful introduction. Thank you very much, Alex, for your very important statements on what patients can do. And I just wish all of you a wonderful week, a wonderful evening, and hope to see you soon again. Bye-bye. For the next time. Bye-bye.
Bye-bye, everyone.